Very good morning, everyone. Dr. Hamid is here. Today we are going to talk about adrenergic neurotransmission, which is only restricted to sympathetic fibers or adrenergic fibers. These fibers, they can release signal molecules which are adrenaline, noradrenaline, and dopamine. And by releasing these signaling molecules, or we can say also neurotransmitter, they can affect target organs. So signaling molecule for adrenergic fibers are adrenaline, which we make it in the adrenal gland, noradrenaline, which we make it in the adrenergic fibers or adrenergic neuron, as well as dopamine, which we have it in different location in the central nervous system. For example, we have it in the CTZ, we have it in the limbic system, we have this in the pituitary gland, and also we have dopamine in the basal ganglia. Today we are going to talk about how we can make these neurotransmitters, how we store them in the presynaptic terminal or presynaptic neurons, and how we can release these, and also we will talk about the different receptors for these neurotransmitters. First, we'll start by synthesis of these neurotransmitters, which we call these neurotransmitters catecholamines also. The precursor for catecholamine is phenylalanine, which we can take this phenylalanine by diet. In the first step of synthesis of catecholamine, we convert phenylalanine to tyrosine by mediation of phenylalanine hydroxylase enzyme. Phenylalanine hydroxylase. This step occurs in the liver. Second step for synthesis of catecholamine is uptaking of tyrosine from external environment into nerve terminal, adrenergic nerve terminal. And this uptake is mediated by the transporter tyrosine sodium co-transporter. This is the transporter which uptake the tyrosine from external environment, and the name for this transporter is tyrosine sodium co-transporter. And this transporter transport one tyrosine and one sodium simultaneously into nerve terminals. Sodium and tyrosine. Next step for synthesis of catecholamine is conversion of tyrosine into dopa. And this conversion is mediated by tyrosine hydroxylase enzyme. Tyrosine hydroxylase enzyme. This step is the rate limiting step. It means that if we can inhibit conversion of tyrosine to dopa, or if we can inhibit this enzyme, we can stop synthesis of catecholamine. We have some chemicals which can inhibit this enzyme. One of the chemical is metyrosine. This chemical, it can inhibit tyrosine hydroxylase and it can stop synthesis of catecholamine. Now think, when do we want to stop synthesis of catecholamine, and what is the indication for metyrosine? We have a tumor which is pheochromocytoma. This is the tumor of the adrenal gland. We make adrenaline in the adrenal gland. If there is tumor of the adrenal gland, we make lots of adrenaline in the adrenal gland. Now we have to stop synthesis of adrenaline in the adrenal gland. This is one of the chemical which we can use for treatment of pheochromocytoma if this tumor is not operable. So the first indication is in operable cases. Also, we can give metirozine before surgery of pheochromocytoma to decrease the level of catecholamines and also to decrease releasing of catecholamines, so we can prevent hypertensive crisis during the surgery of pheochromocytoma. So before surgery of pheochromocytoma, these are the two indications of metirozine. This was about the conversion of tyrosine to DOPA. In next step, we convert DOPA to dopamine by mediation of DOPA decarboxylase. DOPA decarboxylase. Next step for synthesis of catecholamine is uptaking of this dopamine and storing this dopamine into vesicle. The uptaking of dopamine is mediated by a transporter which we have on the cell membrane of the vesicle. This transporter is VMAT. VMAT is abbreviation for vesicular monoamine transporter, and this transporter transport dopamine into the vesicles. In exchange to transportation of dopamine into the vesicle, it excretes hydrogen. Now our dopamine is in the vesicles. Next step for synthesis of catecholamine is conversion of dopamine to noradrenaline, and this step occurs in the vesicles only because we have enzyme dopamine beta hydroxylase in the vesicles. Dopamine beta hydroxylase converts dopamine to noradrenaline. And this noradrenaline is the main neurotransmitter in the adrenergic fibers or adrenergic neuron. And we store this noradrenaline in the vesicles of or nerve terminal of the adrenergic neurons. Also, in addition to noradrenaline, we store some other chemicals in the vesicles. We store ATP, we store chromogranin, we store neuropeptide Y, also we store enkephalin, and all these are co-transmitter which we store them in the vesicles with noradrenaline, enkephalin. This was about synthesis of catecholamine. The first step, conversion of phenylalanine to tyrosine. The second step, uptaking of tyrosine into nerve terminals. After uptaking, we convert tyrosine to DOPA by tyrosine hydroxylase. And this is the rate limiting step. If we can inhibit or if we can stop this step, we can stop the whole synthesis of the whole process for synthesis of catecholamine. In next step, we convert DOPA to dopamine by DOPA decarboxylase. Next step, we uptake dopamine by mediation of this transporter or by the help of this transporter. We uptake dopamine into the vesicles. We convert dopamine to noradrenaline in the vesicles by the help of this enzyme dopamine beta hydroxylase, and we store noradrenaline in the vesicles. Now, in the next step, we will talk about releasing of noradrenaline into synapses. When action potential reaches the nerve terminal, it opens the calcium sensitive voltage channel, and by opening this channel there will be influx of calcium into the nerve terminals. Then this calcium, it mediates fusion of vesicles to the nerve terminal membrane, and there will be releasing of vesicular content into the synapses, in the adrenergic synapses. Now we have released vesicular content into the synapses, and in the synapses we are having noradrenaline and we are having other co-transmitters. This was about releasing of noradrenaline and co-transmitters into the synapses, which is mediated by the calcium. We have some releasing inhibitor and releasing stimulator. The releasing inhibitors, for example, serotonin, it can inhibit releasing of noradrenaline into the synapses. For example, acetylcholine, when it binds to the muscarinic receptor, it can inhibit. For example, alpha-2 receptor of the adrenergic system, when noradrenaline binds to the perisynaptic alpha-2 receptor, it can inhibit the releasing of this noradrenaline into the synapses. But a stimulator for these releasings are when acetylcholine binds to the nicotinic receptor, it can increase. Also, when noradrenaline binds to the beta receptor, it can increase releasing of noradrenaline into the synapses. So the first step was synthesis, the second step was storage, and the third step was releasing of noradrenaline into the synapses. Next step is binding of noradrenaline and co-transmitter to their receptors. We will talk about only noradrenaline because today we are talking about only adrenergic neurotransmission. We have different receptors, different subtype of receptors for noradrenaline. For the first time, Alquist classified noradrenaline receptors into two classes of receptor. One was alpha receptor and the second was beta receptors. So adrenergic receptors, I will write them here. Adrenergic receptors are two in types. One is alpha and another one is beta receptor. We have different subtypes for alpha and beta receptor. For alpha we are having alpha 1 and alpha 2, and for beta we classify them into beta 1, beta 2 and beta 3 receptors. You can see the receptors here. This is alpha 1 receptor, this receptor is alpha 1, this is alpha 2 receptor, this is the beta 1 receptor, this is the beta 2 receptor, and this is the beta 3 receptor. First, we'll talk about the alpha 1 and alpha 2 receptor. We are having different subtypes for alpha 1 and alpha 2 receptor. We can classify alpha 1 into alpha 1A, alpha 1B, and alpha 1D. These are the different subtypes for alpha 1 receptor. And we have different subtypes for alpha 2 receptors also, alpha 2A, alpha 2B, and alpha 2C subtypes. All alpha receptors are coupled with G protein receptor. These receptors are coupled with G protein. This is the G protein and this is the G protein. When noradrenaline binds to alpha receptor, alpha 1 and alpha 2, it can activate the G protein inside the cell, but the difference between these alpha 1 and alpha 2 receptor is that they are coupled with different type of G protein. The alpha 1 receptor is located post-synaptically. But alpha 2 receptor, it is located presynaptically, it means in this membrane, in this neuron, and also postsynaptical neuron. So the location for alpha 1, postsynaptical neuron, and for alpha 2 we have presynaptical neuron and postsynaptical neuron, as well as we have alpha 2 receptor in the pancreatic gland also, the pancreas also. This was about the location of alpha 1 and alpha 2 receptor. Another difference between alpha 1 and alpha 2 is that alpha 1 is coupled with GQ protein, but alpha 2 is coupled with GI protein. When noradrenaline binds to alpha 1, it activates the GQ protein, and now all you know about it, GQ protein will increase the concentration of calcium in the cytoplasm of target organ, and by increasing the calcium we can see increasing in the contraction of the muscle and increase in the activation of different enzymes in the target cells. So this will increase the GQ, and this GQ will activate the phospholipase C enzyme. Phospholipase will converts phospholipid into DAG and IP3. IP3 increase releasing of calcium from sarcoplasmic reticulum, and this DAG activate protein kinase A. Alpha 1 receptor, the location for alpha 1 receptor, we have alpha 1 in smooth muscle cells, we have these receptor on the membrane of smooth muscle cells. When noradrenaline binds to this alpha 1 receptor on the smooth muscle cell, it increases the calcium, and by increasing the calcium it can increase contraction of the muscles. As well as we have this in the smooth muscles of the prostate gland, and we have in the smooth muscles of the vessels. The agonist for alpha 1 receptor is, all we know that phenylephrine is the agonist for alpha 1 receptor. I will write it with red. Phenylephrine is the agonist for alpha 1 receptor. And the antagonist for alpha 1 receptor is prazosin. This is one of the antagonists for alpha 1 receptor. About alpha 2, when noradrenaline binds to the alpha 2, it activates the GI protein. This GI protein decreases the cyclic AMP. It can decrease also, it can block the calcium channel also, and by blocking the calcium channel, it prevents releasing of noradrenaline and other neurotransmitters into the synapses. This is the other effect. We have alpha 2 in the pancreas also. When noradrenaline binds to the alpha 2 receptor, which is on the pancreatic cell, it can inhibit the releasing of insulin. The agonist for alpha 2, I will write it again by red marker. The agonist is clonidine, and antagonist for GI protein is euhimbyne. These are the agonists and antagonists for alpha 2 receptor. About beta receptors, we have three subtypes of beta receptor: beta 1, beta 2, and beta 3. All these three subtypes are coupled with GS protein. And when noradrenaline binds to these three subtypes, it can activate the GS. By activation of GS, we can see increase in the cyclic AMP inside the cells. Also, they can open the calcium channel and they can block the potassium channel. We have beta 1 receptor on the heart. We have beta 1 receptor on JG cells, juxtaglomerular cells. When noradrenaline binds to beta 1 receptor, it can stimulate or it can increase the heart rate, it can increase force of contraction, it can increase cardiac output. When it binds to the beta receptor on the JG cell, it can increase releasing or secretion of renin, which this renin will convert angiotensinogen to angiotensin first or angiotensin 1. The agonist for beta 1 receptor is dobutamine. Dobutamine is the agonist. And antagonist for beta 1 receptor is metoprolol. Beta 2 receptor, we have this receptor on the respiratory cells, on the lungs, and some other organs also. For example, we have this receptor in the vessels, we have this receptor in the pancreas, and by binding of this noradrenaline to this receptor, we can see different effects. The agonist for beta 2 receptor is terbutaline, and antagonist for beta 2 receptor is propranolol. And about beta 3 receptor, we have this receptor on the bladder as well as lipocyte. When noradrenaline binds to the beta 3 receptor on the lipocyte, it will increase lipolysis. And when it binds to the beta 3 receptor on the bladder, it can relax the detrusor muscle and it can decrease the urination. This was about the effect. An agonist for beta 3 receptor is mirabegron, which we can use for treatment of overactive bladder, as well as some other agonists for the beta 3 receptor, because they can increase the lipolysis. We can use them for treatment of obesity. This was about the receptor as well as agonist and antagonist of each receptor. But after binding to the receptor, the next step is terminating the action of noradrenaline. We can terminate the action of noradrenaline by different strategies. The first strategy is uptaking of noradrenaline by a special transporter, which we call it NET, norepinephrine transporter. NET is abbreviation for norepinephrine transporter. This transporter, it uptakes the noradrenaline and it transports noradrenaline from synapses into the nerve terminals. When this noradrenaline comes into the nerve terminal, either we can uptake this noradrenaline again by VMAT and we can store it into the vesicle, or this noradrenaline, we have a special enzyme which is MAO, monoamine oxidase. This enzyme binds to noradrenaline and it metabolize the noradrenaline to its metabolite. Metabolite for noradrenalines are metanephrine, normetanephrine, we have VMA, which is vanillyl mandelic acid, and we have hydroxy mandelic acid. These are the metabolites for noradrenaline. So the first strategy for terminating the action of noradrenaline is uptaking of noradrenaline with NET or norepinephrine transporter. It uptakes the noradrenaline into the nerve terminal. This noradrenaline, which is now in the nerve terminal, we can terminate the action of noradrenaline by the help of or by this enzyme, monoamine oxidase, or we can uptake it again and store it into the vesicles by VMAT, vesicular monoamine transport. The next strategy for terminating the action of noradrenaline in the synapses is by mediating of this enzyme. This enzyme is COMT, catechol-O-methyl-transferase. This will bind to the noradrenaline in the synapses and it will convert noradrenaline into its metabolite, and it will terminate the action of noradrenaline. Then we distribute these metabolites to the liver. In the liver, we conjugate these metabolites with sulfate and glucuronic acid. And by conjugating with sulfate and glucuronic acid, then we can excrete them in the urine. After conjugation, they are water soluble and we can excrete them in the urine. This was about the terminating of action of noradrenaline. But if this adrenaline or noradrenaline is in the adrenal gland, we can terminate the action of noradrenaline and adrenaline in the adrenal gland mainly by the help of this enzyme called Untitled Chapter. Another strategy for terminating the action of noradrenaline is uptaking of these noradrenaline by adjacent neuron. We have one transporter which is ENT, extra neuronal transporter. This extra neuronal transporter is located on the adjacent or neighboring neuron. It uptakes the noradrenaline, and by uptaking the noradrenaline, then we can see either storing of noradrenaline in the vesicles, or again terminating of the action of noradrenaline by MAO, which is in the presynaptical neurons. So for terminating the action, uptaking of the presynaptic neuron, uptaking of noradrenaline with ENT of the adjacent or neighboring neuron, or terminating the action by alcohol or ethyl transfer. This was about the adrenergic neurotransmission lecture. In this lecture, we talked about the synthesis, storage, releasing adrenergic receptors, as well as terminating the action of or metabolism of alcohol amines.